Hello, everybody. It's me, Raven Baxter, also known as Raven the Science Maven, and you are watching STEMbassy. And I'm so excited to be here tonight. It's our second last episode of the season, and we have a very special guest on here tonight. And if you guys are tuning in to watch STEMbassy for the first time, let me know where you're watching from. If you're in the chat, make sure that you let us know where you're watching from in the chat. We are looking in the chat. We love to read your comments and hear what you have to say during the show. We like to engage with you all, so don't be afraid to say hello because we love hearing from you. This is the place where STEM ambassadors meet. It's called STEMbassy, and that's STEMbassy, which stands for STEM, Science, Technology, Engineering, and Mathematics. And this is a weekly talk show centered around STEM. It's hosted by 5 incredibly talented and passionate women in STEM, and you're about to meet all of us very soon. Every week, we bring on a STEM professional to highlight and interact with. We learn about their STEM story. We talk about things that they do, things that they're passionate about. And we also talk about current events, hot topics going on in our world as they pertain to our culture, within STEM and beyond. And then we also play games, okay? We have a good time here. This is not a boring STEM show. I don't know what you heard about STEM, but this is fun STEM. This is where fun people hang out. I was gonna say the fun kids. No, we're grown folks. We're grown folks over here, but we're fun. And every week we come on here. So without further ado, I would like to introduce our co-host on the show. So it's time to hear from Jay, Amanda, Chanel, Ariana. Hi, Carla from the audience. Hello from Bethesda, Maryland. Thanks for watching. And hello to Toya. Thank you for watching. Another fellow STEM ambassador in the audience. Okay. Jay, you ready? Hey, Jay.
Yes. Hello. Actually, Raven, we could say that we're the cool kids because we are.
Okay.
But so
I'll take it.
hello, everybody. My name is Jay. I'm a UX researcher and international psychology student who is currently in Kuwait in my field experience. So pray for me with my time and sleep. I am interested in AI, cybersecurity, and women empowerment. I'm also the host of a podcast called Get Into It Podcast, where I unpack subjects of culture and technology from a psychological standpoint, and I release episodes every Tuesday. So make sure you guys follow me there on all socials at Get Into It Podcast.
That's right. And one thing you'll hear about or notice about all of our hosts is that each of us have our own little thing going on, okay? And they're not even little things, they're actually big things, okay? Big things popping. All right, so make sure you check out Get Into It Pod. Jay is amazing. And can you tell us what time zone you're currently operating in and why?
Yes, Kuwaiti time zone. So I am in my field experience because I'm about to graduate and get this master's in international psychology, praise God. So shoot, now I'm trying to remember my CST time. So I'm supposed to be here at 7, but it's like 3 a.m. in Kuwait.
Oh my gosh. Wow. Mm-hmm. So Jay's working overtime basically to hang out with us. So we appreciate her time. And we're definitely going to make sure it's worth her time because we have a good time here on STEMbassy. All right. Well, speaking of a good time, let's bring in Amanda.
Thank you. You know what it is. Hi, everybody. I'm Amanda. I'm a PhD student at the University of California, San Diego. My lighting is very weird after daylight savings. I do apologize. And I also have a podcast. It's called Triplicates Pod. We are 3 first-gen women of color going through a PhD program, trying to figure it all out. And we're going to share our experiences, tips, advice, and talk to some folks about all the things that you don't have to learn the hard way. So you can check us out on Instagram, Twitter, and most streaming platforms at Triplicates Pod on every other Thursday.
Love it.
Thank you.
Oh, and we have some fans.
Yes. Okay, Queen.
Fans in the audience. Love Triplicated podcast. Love it. We know what you meant, sis.
The autocorrect do be getting you sometimes.
They do. We appreciate it anyways. Wonderful. And so let's bring up Chanel. Hey, Chanel.
Hey, y'all.
Hey.
Hi, I'm Chanel, also known as the Public Health Bae. I am a cell and molecular biologist with a doctorate in Public health, you guessed it. I presently work in human health and toxicology. From time to time, you'll see me go like this and put on my public health hat to give you some insight as to how some of our STEM topics relate to cultural competency, health education, and health literacy. Today's public health hat is going to be kind of funny, so stick around. Welcome to STEMbassy.
We love to see it. And she's got that nice quarantine glow. Skin's looking real nice.
There's a glow.
Hey.
We love to see it.
This is what happens when you follow directions and stay away from people. This is what happens.
Stay inside the house. Yep.
Oh, love it. Yep. Okay. And then last but not least of our co-hosts, we have Ariana. Sorry, December.
Oh my gosh, guys. Glad I finally got to wave. Chanel, when you came on the screen, I waved at you and realized you couldn't see me. I don't know why, but I did. I'm Ariana. I'm known as Ari the ecologist on the internet. I'm a wildlife biologist, and I just finished my master's in wildlife ecology studying bobcats in New Jersey. I'm currently an adjunct instructor and a wildlife technician, but I will be starting a PhD position in like a month at the University of Idaho studying wolf ecology. And so I'll be starting that in May, which is crazy. I also run an Etsy store called Wildlife Wanderlust, where I sell prints of wildlife from the field and other fun wildlife-based art. So you can check out things like this cool coaster. So you can buy things like this. This is currently up on my shop. Make sure to check it out. And I would also like to shout out December, who is our amazing producer and also part of the STEMbassy team. If you hear us talking to her, just know that she's working hard to keep our stream running smooth. Also, make sure you are following STEMbassy on Twitter and Instagram at STEMbassy, on Facebook as The STEMbassy, and you can find links to all these platforms at our website, stembassy.org. And don't forget to join us on Patreon at patreon.com/STEMbassy. So I hope you guys caught all that. Make sure to go and give a follow.
Yay! We love it. And then Ariana, where can we find? They could just search for Wildlife Wanderlust.
Yeah, that's the name of the store. No space when you're searching it on Etsy. You can also go to my Instagram at Ari the Ecologist, and in my link in the bio, you can also find it.
Okay, wonderful. We love it. And again, this is our second to the last episode, so I'm like a little sentimental because
3 seasons almost done.
3 seasons, over a year.
Wow.
We've been doing this for over a year, everyone, all of us together. And when I tell you this is a solid team and we put the work in, everybody is just so amazing. So I feel very fortunate to be working with y'all. And this show is amazing because of everybody here on the stage. So give a round of applause. Let's just clap for ourselves. You know what I'm saying? It's a big deal. We're doing big things. We work hard. And another thing that we're especially excited about tonight is our guest, because this is somebody who, well, for me, okay, I'm just going to talk about why I'm excited. This is somebody who I really admire in the STEM space. This is a science communicator, somebody who does work that is very similar to what we all do and what we're all passionate about. But, you know, she's going to come on and share her story so we can learn more about her. This person is named Imogene Cancellare. They are a conservation biologist and PhD student studying snow leopard genetics. As a science communicator, she works to increase public knowledge of wildlife issues by making nature accessible and interesting to everyone. Join me in welcoming Imogene Cancellare. Hello.
Hey, everyone. It's awesome to be here. I'm super pumped.
We're pumped too. Welcome to STEMbassy.
Excited to have you.
Yeah, no, this is super cool. I am like, the energy is fantastic. Like chatting backstage, you guys are fantastic. So I'm super looking forward to this. It's going to be a fun hangout session, but also we totally hang out in real life. So
Absolutely.
Let me know when you're in the California area.
Absolutely.
We should all meet up there.
Come through, y'all.
Yeah,
We
so
could
excited.
get back, you know.
I think
Social
we can-
distance, walk.
we've been really fortunate to have like actually more than one wildlife ecologist on our show. I feel like we've went above and beyond improving our selection. Like I'm really happy to have you on and learn from you today.
Yeah, no, I'm always down to talk wildlife, and it's super cool that you guys are like featuring all the wild critters and the people who study them.
Woo, woo.
Yeah, that's awesome. Well, before we get into our conversation, I do want to highlight that we are raising money for Backyard Base Camp this week. That is our charity highlight of the week. And so Backyard Base Camp was founded and is operated by Atia Wells, a pediatric nurse with a passion to introduce, educate, and connect families in Baltimore, especially those of color, to local outdoor spaces. This organization inspires Black, Indigenous, and all people of color across Baltimore City to find nature where they are and empowers them to explore further. So this is a great cause. So make sure you check out their website at backyardbasecamp.org. And Imogene, you selected this as a cause that we should be supporting. Is there anything you'd like to add about this organization?
Yeah, no, I mean, I just think Backyard Base Camp is doing really cool stuff. I became aware of their work and the work that Atia is doing sometime last year because of Twitter, like so many things. And I'm just really passionate about people getting outdoors in whatever way feels good to them. I had the privilege of spending part of my childhood on a farm, and not everybody has that privilege. And certainly when you look at different wildlife spaces, you see a lot of the same people over and over again. And so whether it is bringing someone to wildlife or to the outdoors as an adult or as a kid, I think that's something that is really awesome. And the fact that they're doing that here in Baltimore, around in the Baltimore area, is not only showing people that anybody can have access to wildlife, but anybody can enjoy wildlife. And so I just think that's super solid.
It's awesome. I was looking at their website and I saw that they had a gardening initiative. And I know Chanel, can you give us an update on your garden? I love hearing about your garden.
Yes, I can. So for those of you that have been following along since last season, I started a project called Let's Grow Together. And so the goal for Let's Grow Together is to show people, particularly Baltimore City, we're on the topic, who live in a food desert or a food swamp and don't have a lot of land available to them to grow their own gardens, to start a container garden. So you can do this in the backyard on like your little plot of cement. You can do it in your balcony of an apartment complex, but you can have a container garden and grow some high yielding crops that will help extend your meals. And you can cook fruits and vegetables. I did melons. I did various veggies that are high yield, like tomatoes and zucchini and cucumbers. So those things are really high yield. And you can make salads. You can saute them and add them as a side vegetable. There are so many things that I was able to do last season, and I'm starting again this season. And service announcement, if you have not already started, if you're in the Maryland area or in the DMV, you'll notice that we've had an extra long frost season. So if you have not started your garden yet, it's okay. Go ahead and start your season inside now, because when the weather does finally break and gets really warm, you'll be able to transplant them. We can get started. Let's grow together.
Right.
So get ready.
That's awesome.
Thank you.
Exciting. And so, yeah, check out Backyard Base Camp. They also have some gardening information. So that would also be a great resource.
Thank you so much for sharing that with us. We love bringing on people who have social causes, because that's what we're all about, is impacting our communities and making sure that we stand for something in addition to science, because science is amazing, STEM is amazing, but if we're not also involving our thoughts with our communities, then what are we doing? So wonderful. Yeah, we're definitely going to make sure we bring awareness to Backyard Base Camp for you. But most importantly, we do want to learn about you, because you're the star of our show today.
I have a question.
Yeah.
You mentioned that you grew up on a farm, and now you're a conservation biologist. I feel like those two things are connected. What kind of animals did you have on the farm? How did you get into like, okay, this is what I want to do. We're saving the animals. Like, how did we get here?
All right. So I grew up on a dairy goat farm. we like where you would milk the goats. We didn't like make cheese and stuff, but like we'd mostly sell our milk to like hippies who drank goat milk, but also for like any other commercial product. And I, you know, this is in like rural North Carolina, and so like we had dairy goats and we had like chickens and sometimes pigs. We had like sheep and cows and donkeys occasionally. And I always wanted to be a veterinarian. Because, you know, if you like animals, you like being outdoors, it's like, oh, great, you know, you are obviously a veterinarian, because that is I guess the monolith of someone who likes animals is a veterinarian, which is not actually the case, but I didn't know that for a very long time. And so when I went to school, I majored in animal science, and I was taking like pre-vet courses. And I did a study abroad I'm dating myself, in 2008. We went to Australia, and we were looking at the livestock wildlife interface, and how Australia manages their livestock, both in terms of like husbandry and management and the science behind that type of production, but also the unique interface with like a more wild landscape. So like picture like a whole 1,000 acres of like rangeland with cattle on it, but also like there's a herd of kangaroo running through. So totally different from what I was used to. And we were going back and forth between these like lectures on like wildlife and lectures on like food animal production. And the day that it happened, we were outside doing this exercise with, what were we doing? We were like dehorning a cow. We were doing like a surgical procedure. And we came back in, we all filed back inside to sit down, and we had this lecture on coral reef die-off. And how it affects different systems, both like people who rely on that type of ecosystem, but more specifically all of the different wildlife and the trophic cascades. And I just remember sitting there, and I had this paradigm shift. Like my entire life had been leading up to trying to go to vet school. And all of a sudden I just realized I don't want to do that anymore, because there's so much more out there than just being like a veterinarian. Not to knock vets, like obviously like it's a cool field. It's, really difficult to be a veterinarian. And I just realized that I wanted to, combine my interest in animals and like my fascination with science in a totally different way. And for me, it was like, oh yeah, conservation. Like, how can I be useful for something that's bigger than me? So like that was the whole coral reef thing, like this big magical moment. I was like, oh, something bigger. This is what I can put myself towards. That's bigger than me. But how can I also do it in a way that like will be useful? Like, what skill sets do I have that can actually be valuable in the future? And so a lot happened in between like then and now, but basically that's kind of put me onto the path that I'm at. So basically goats have nothing to do with what I currently do, other than the fact that the things that I study do like to eat goats. That was a long answer, sorry.
That's totally fine.
That's such an important story though, because I feel like from my experience, just talking to first year students in science programs, often they, obviously your freshman year of college, you have to select a major for many schools. For some schools, you can go undergraduate for some time. But for all of the schools that I attended several, you had to choose a major your freshman year. And we don't really be knowing, if you haven't had the privilege of having multiple career exploration experiences, you kind of just go with what you know best and what you have the most exposure to. And usually those are those mainstream careers like vet, doctor, firefighter, lawyer, and so I think a lot of students kind of get down on themselves when they get to that point that you spoke about, Imogene, when they're like, when they realize, oh, I actually don't want to be a vet. I actually don't want to be a doctor. But some people see that as a failure, like, oh my gosh, like, I failed, but you took it as a moment and you're like, okay, time to change my path, like time to change what I'm doing because I'm finding my passion. And I think that's really important. Like, it's okay to change your mind, even when you're in the middle of something. As long as you're following your heart, I feel like you can do no wrong. Well, how do you guys feel about that? Have you ever been in those moments?
Often? Well, not necessarily now, because I think we all can agree we found our voices. We know exactly what we want to do. But when you're trying to figure it all out on what area you want to pursue, it is kind of hard to step away from tradition and not feeling like I'm going to disappoint somebody because I'm not going this way. And then, how it's presented to us, you should be a doctor. You should do this because these are the natural steps. And it's not, because there's so many other ways to, explore the things that you're passionate about and that you like. We just may not have the, not the resources, but the exposure to them. So yeah, I mean, I feel all of that. All of it.
I definitely agree. I think we bring a lot of people who are PhD or PhD candidates, or they've passed their PhD exams, and they were kind of like, oh yeah, I came in pre-med because I didn't know what else there was. I was like, oh, you like biology, you are a doctor. Oh, you like animals, you go be a vet. So it's definitely interesting to hear like the flip side of that on like more of the animal side of like, okay, yeah, it's not just us that are trying to figure out what options we have as a career path out there. It's like happening in a lot of different fields where there has to be a push of exploration of options. And that's why we bring people on here to talk about like, this is something you can do and it's totally fine and it's totally fun. And I'm very happy to have you on. So thank you for coming.
Yeah.
Amanda, you even have crazy people like me who were biology and pre-med and then did a complete 180 and hopped all the way over to wildlife.
That's true.
Yes, we love all paths to science. It's fine.
Yeah, they are definitely fun.
Yes, and it's important.
Oh, go ahead. I'm sorry.
I was just going to say it's important to know that the path is not always linear. We've always talked about this. It's been a frequent occurrence or a topic on the show. The path in STEM is not always linear for everyone. So don't look at that as something that's not an accomplishment or that's a deterrent. That's just the way it is sometimes. It's not always linear.
Well said.
Absolutely. So I have a two-part question. So for you, Imogene, can you explain why you had to dehorn a cow? That's the first time I'm hearing of it. And then two, I'm trying to picture, because everybody else on the show knows that like I didn't grow up on a farm, but I grew up in the country, and I now want a farm, but I want it in a city. I'm not for sure how that's going to work. Whatever, we'll figure it out. Either way, how is it being in this field as a woman? Do you ever get like, just hold this for me and just observe, or if that's a word, I can't.
Yeah.
Think. But yeah.
So to answer your first question, so when you're dealing with livestock, so let me back it up a bit. So like goats and cows are different in terms of their horns. Cows have really hollow horns, like literally think like a glass. Like they have some vascular structure in them, but they're not like this hugely innervated thing coming out of their head. Whereas like goats, as they get older, they have a lot more veins and they're a lot thicker. And so you would treat goats totally different. But in the case of cattle, because they don't have these huge vein systems, you can basically remove their horns without harming them. Generally in livestock, if you want an animal to not have horns, you can either breed it genetically so it doesn't have those traits, or you can do like what's called disbudding when they're really young, and it's like literally cauterizing the tissue where those little horns are going to grow up. So basically the scar tissue prevents it from growing, because you basically like, I guess, just burn all the cells off. Like that sounds kind of gross. But in the case of what I was doing, sometimes, I don't really know why, you would have an animal that was older that either somehow accidentally skipped that step, like you didn't catch it, or you did the cauterizing step and it still grew. And so what we were doing is we literally had like metal floss, and you basically wrap it around it, and you were like trying to like saw it off. So you're using like this metal floss, and like the friction from pulling back and forth basically slices it right through. And in the exercise we were doing, there was like there was no blood. Whereas if you did that on something like a goat, I mean, it'd be a hot mess. And so we were doing this in that case because the animal, I think that they just missed it. And it was like 2 years old and it had these horns and they wanted to remove it. So it was a teaching exercise.
Is there a reason for removing it? Like, does it hurt? Does it hurt an animal?
So in this case, for a cow, the only discomfort they would have felt is the fact that they're just kind of hanging out, and someone, like the pressure of like someone moving their head around. But because like the horn itself is keratinized, it would be like cutting your nails. Whereas if like you couldn't do it with a goat, like once you got through the keratin of the horn, there's so much tissue down in it, like that would be like an egregious thing. So like you would never do that. The disbudding is like short term, not great. Like it basically be like someone taking like, I don't know, like a cigarette lighter. And you obviously like you can burn your hand. And so it's the same thing right here, but because goats have such really thick skulls and because they spend so much time like fighting with one another, it's not as, at least we don't think it's as sensitive as if someone like took a cigarette like lighter and like, singed our skin. And so
is this something that we could pull up a picture of for reference, or is this something we should not be pulling up the picture for reference?
Maybe, probably not.
No? Okay.
Probably not.
No. Okay.
That's the weird thing about agriculture is, there are like standards of things that you have to do.
That was the question I was trying to get at. Like when I asked about does it hurt the animal, what I meant to ask was the reason why you're doing it. If you don't do it, will the animal end up hurting themselves? Like what would be the reason to dehorn? No, it's my fault for not clarifying. I'm so sorry about that.
And no, that can certainly be the case. I think more often than, there's two different reasons more often than not. One is when animals are in small enclosures together because they're territorial and they have a hierarchical system. Animals that have horns are more likely to fight. And so because you're basically changing some of their natural behavior, you can introduce like harm between between two individuals that are fighting. But because they're also in fences, yes, animals can, they can get stuck in fences, and that can, involve like broken limbs or in like worst case scenario, like breaking their neck.
All right, Imogene. Now we're talking about livestock and goats and cows, and I know that you study snow leopards because I was in the same lab with you for 2 years. And snow leopards happen to a lot of times like to try and eat goats. You said that earlier, right?
Yeah.
Can you talk a little bit about your research in general and maybe tell us a little bit about what you're doing with your snow leopard research?
Yeah, absolutely. So I am currently in the 5th year of my PhD at the University of Delaware. Ariana and I were in the same lab. I am still there. I am currently focusing on range-wide, so a range-wide phylogeography and genetic structure of snow leopards. And so what that means is we are trying to paint a picture of the molecular puzzle of snow leopard populations throughout their entire range, which is going to be 12 different countries in Central Asia. So starting off with, if you can think of a map of Central Asia, or if you can't, that's totally fine. If you starting off at, like, let's go with the clock metaphor. 12 o'clock is Russia and Mongolia. Moving around to like 9 p.m., you've got a lot of the different stands like Kazakhstan, Kyrgyzstan, Tajikistan, Pakistan. Snow leopards are in those countries. Curling down around to 7 o'clock, you've got Pakistan, and then you're moving down to 6 o'clock, you've got Nepal and Bhutan. And then you've also got in between that, kind of in the center of the clock, you've got China. And so snow leopards persist in 12 different countries. I don't think I missed one or two, but I got close to 12 throughout the high elevation mountain ranges in that part of the world. So that's anywhere from like 4,000 to 20,000 feet in elevation. And we know that snow leopards like rough, arid, rugged terrain. They live at high elevation. They're high montane specialists, but there's a lot that we don't know about snow leopards relative to other charismatic species or just other species in general. And so my research is focused on trying to understand their population structure. So what I do is I am combining fieldwork and lab work to genetically identify individuals, and then basically lay them down on a map and create a genetic map of their population structure so we can understand snow leopards in Kyrgyzstan and Tajikistan are one population, and they're really well connected to snow leopards in Pakistan, but they're not well connected to snow leopards in Russia, and so on. And this isn't real data. This is just me kind of hypothesizing as I'm explaining it. The idea is that if you understand where animals move and how they're connected, like through gene flow, you're understanding their movement, but you're also better able to predict things like future connectivity and change to different like environmental stressors or landscape variables. In particular, climate change is something that is obviously important everywhere, but Central Asia, these high elevation habitats are warming at a faster rate than other parts of the world. And so when you're dealing with an animal that literally lives on the roof of the world, in a snowy environment, there's only so many places it can go. So some of these questions that I am asking are with the goal of trying to kind of inject genetics into our conservation toolbox.
Really cool. And where are you getting the genetic samples to be running these kinds of like gene comparisons?
I'm glad you asked that. So my research is non-invasive, which means that I don't ever have to come into contact with a snow leopard, which is great because snow leopards are really rare and elusive. And also it would take me a really long time to get enough data from individual cats. Non-invasive research means like you don't have to come into contact with the animal. So like that might be camera trapping. We're looking for like tracks and sign. But in my case, because I want genetic material, that means I spend most of my time out in the mountains looking for cat poop. Because you are able to get genetic information about who pooped, or from the animal who pooped, because when an animal defecates, they shed off epithelial cells from their intestines, and within those epithelial cells is your DNA. And so we go out into the field and we collect that and we bring it back into the lab and we extract DNA from the outer casing of that material, so we can hopefully amplify enough of the target DNA, so the animal who defecated, so we can identify not only species but then also identify individuals.
I have a question, because I've spent years of my life basically synthesizing poop in a lab and then extracting the DNA from it, specifically E. coli. But that was like they were all clones of each other. And so I'm wondering, when you're dealing with actual poop from an animal, how do you isolate the epithelial cells, meaning the skin cells for everybody watching, or like the skin cells, tissue cells, from the bacteria, from the plant matter, from like everything else that's in the poop?
Okay, that's a great question. So first of all, before I answer it, I just have to say, Raven, we should have a conversation, because one of the things that we are trying to do is amplify fecal DNA to use SNPs, which are single nucleotide polymorphisms, which is a molecular marker that has a lot more statistical value than some of the other markers we use. But the problem is that fecal DNA is not always great. And so Raven, if you've been doing stuff like that, we should talk, because that is the golden egg in conservation genetics. And if you can help me solve it, that'd be great. So Raven brings up a good point. So once an animal defecates, like the poop drops to the ground, microbial processes start, the decaying process starts, everything just crawls out of the ground because they want some of that good stuff. In most environments, you have a really short window in order to get to that sample before the DNA is going to be degraded, both from the microbial activity, but also from UV light. The benefit about the work that I do is that snow leopards are at really high elevation, so they're above tree line, and it's relatively dry all the time, and it's, for the most part, relatively cool, if not very, very cold. And so dryness and coolness are two good things when it comes to preserving DNA. And so there's very little microbial activity. I mean, obviously there's going to be some, but there's not significant microbial activity to break the scats or the fecal material down very quickly. And so we have scats that can persist on the landscape for like a year or two, and you can still actually successfully extract DNA from them, which is cool.
That is really cool.
In the way that we do it, I'm just thinking about I need something to explain it. Here we go. All right, I got a candlestick here. So you asked the question, so if anybody who doesn't know, we're gonna pretend that this is, just roll with me, guys. Just pretend that this is poop. Like something just dropped it on the ground. If I'm collecting it for my research, I'm not taking the whole thing. I don't need all this. I don't need this baggage in my pack. I don't need this much. So depending on your research question, you definitely obviously want to take enough. And so there are certainly things that we can study, like diet. And so you would want to take a decent amount of the fecal material. But the way that we maximize the likelihood of getting our target DNA being whoever pooped this, is we shave off the outer layers of the scat material. So people might think, if you're looking at it this way, oh, well, just take a cross section of it, take a little Tootsie Roll, take it in the lab, grind it up. No, that's too much, because you're going to have a lot of your prey substance, or your prey DNA, and undigested material, and whatever else like came out of the body. I'm interested in the stuff on the outer edge, which yes, can also sometimes include things like dirt and grass that was on the ground, or it can include prey DNA as well. But once we basically take the outer casing of the fecal material, we use species-specific primers to amplify what we're looking for. Which, you know, helps us as opposed to like just taking like giant cross sections of it. I can talk about poop with literally anything in front of me. Today just happens to be a candle.
I appreciate it. This audience member, I actually don't know how to pronounce this. Josue Arteaga Torres mentioned non-invasive research in wildlife conservation's rules. And I think this kind of goes along with the conversation I've been having online today about what science professions can look like and what the stereotypes of doing our job in science can look like. And that, you know, I'm sure you might have had this issue where, you know, somebody really cares about you, loves what you're doing when they hear about you being a scientist, a biologist, and that you work with animals. But then they're like, okay, let me see some animals. Show me some animals. Give me some pictures of you with animals.
Right.
And there's this just misconception that our work looks one way when the reality is, you know, you can be a conservation biologist and be doing data science and working with sequences of genes and proteins, and like that kind of work is still conservation biology. And so, you know, when this person made this comment about wildlife non-invasive research ruling in wildlife conservation, can you talk about maybe the importance or why it rules and why they think it's so awesome and what does it mean for you?
Yeah, no, the reason I think it's great is because obviously there's so many different ways that we can get at a scientific question or like analyze some metric of a wildlife population. And certainly there are relevant questions that require live capture and handling animals. So maybe you're looking at like, and you know, looking at like gut microbes and looking at like endoparasites, or maybe you are trying to track, you know, population movements, or, you know, understand how they interact with their landscapes. So you need to live trap and put a radio collar on an individual. But certainly even with some of those questions, there are ways that you can do it without ever coming into contact with the animal. And so the benefit of that is twofold. And I think the most important thing is not about the science. The most important thing is about the fact that no matter how careful you are, interacting with wild animals is always going to be very stressful for the animal. And so, yes, we have to get permits, but even with all the permits and all the training in the world, like I've handled lots of wildlife under, you know, I've been the lead person, I've assisted with other things, I've had lots of training. And so I'm certainly comfortable in being able to do that. But ultimately, at the end of the day, that animal is still going to be physiologically stressed out, and that could impact their survival in some way. And so if you're able to minimize that, I think that's important. But then also, the cool thing about non-invasive techniques is that you're able to answer so many questions that you wouldn't be able to answer by capturing and handling individual animals. And so, like, I'm studying genetics, and I think genetics is awesome, but the cool thing about it is I am going to be able to understand how vast numbers, or at least as many as I've sampled, understand how animals are connected across the landscape. So it's not just understanding how they're related, but it's understanding how they perceive the environment that they live in, which tells us so many more things in other different disciplines, which can help like jumpstart other relevant questions for their conservation. So someone else can come and, you know, answer a question that they're interested in. And so I think it's really just like, it's really limitless. I mean, yeah, handling animals is super cool, but like, you know, pulling DNA out of poop is really cool too.
Yes, I love that for you. We have a question. Yeah, I'm just the poop thing, and it's funny because I have a question from Asama, and Asama also put in the chat that she loves poop, and you study human poop. So like, I feel like you guys would really vibe. But she has a question about how do you figure out population structure, like a genetic tree? Can you explain that process a little bit?
Yeah, so there's a couple different ways. So there's two different ultimately there's two different populations, and the difference is based on time. So when you're talking about trees, you're talking about at least in my experience and what I do, you're talking about phylogenies, which is basically tracing genetic lineages through time. And so in the case for most species, we do that using mitochondrial DNA. Mitochondrial DNA is maternally inherited. It evolves really rapidly. And so as a result, you're able to trace like generations throughout time. And so we use markers from mitochondrial DNA to get an idea of how several species are related, or how within an individual species, how different populations are structured based on like haplotypes, or basically a haplotype is a certain amount of genes and DNA that are inherited together. So it's kind of like a bookend, or like a blockchain of things that are inherited at the same time. And so that tells us a lot about how individuals are related, and that gives you an idea of like their population structure. Whereas if we're talking about something more current, like contemporary population structure, we use we use molecular markers that are generally not subject to the constraints of adaptation or evolution. So we use neutral markers, so they're not under like positive or negative or sexual selection. And this gives us an idea of just how gene flow, whether it's like genetic drift, or basically like animals are isolated, you basically are able to get a picture of how they're related. And the way that we do that is, after you're done in the lab, you take the data and you can plug it into these like Bayesian statistical algorithms that then basically compare all their similarities and basically pop out a map to show you like clusters of potential populations. Sorry, my cat's like yelling in the background. Apparently I didn't explain that very well.
Join the club. Imogene, I have a question. When you talk about inputting the data into the software packages, right? How do you use that data? So you got the DNA, you've extracted it, you've loaded it into the programs. Now what?
Right. So once you get your raw data, so raw data being like all of the wet lab work that you do to determine like, okay, I've got like 50 individual snow leopards from this part of their range country. I really want to understand a lot of descriptive statistics. So I want to understand what is the balance of the different alleles that are in these populations. I want to understand whether or not they are in or out of Hardy-Weinberg equilibrium, which is a series of requirements that tell you if a population is under certain types of constraints, like genetic drift or non-random mating. And then once you get like that kind of like basic, that's like your bare minimum, like this is what's going on with the genetics, then you can start playing around with that data. And that would include things like looking at their population structure, which I already described, or looking at things like connectivity. And so I don't know if Ariana's talked about some of her work. She's done basically connectivity research with her bobcats. It wasn't genetic, but you can do that same type of work by inputting your genetic data into these programs that then manipulate it compared to like environmental matrix, or compared to like something that you care about in the landscape, like the geography, or, you know, you have your map of, we know there are two populations. Is there a reason that there are two populations? Is it because there's like a river running in between them? Is it because historically they weren't connected? Is it because they're under like their experience in evolution, they're becoming two different species? Questions like that. Often like these analyses lead to more questions. And I haven't gotten to those yet. I'm currently still finishing up my lab work, so I don't have any results yet, unfortunately.
That's awesome. And yeah, connectivity is cool. I like connectivity. Mainly because I've spent a long time doing it. So you mentioned that you have both wet lab work, kind of data analysis work, but you also got to go in the field to collect poop. So what are some of the craziest experiences that you've had in the field, either with your snow leopard research or even before working on other projects?
Yeah, so I was really fortunate to be able to go into the field in 2019. I've been really fortunate, really fortunate. I feel like that's important to say. Like, I feel very lucky to be in this PhD. It's truly a privilege to be able to study this species, but also be able to, I'm doing international research. And so I feel like I frame everything from the perspective of I'm a guest. Here in the United States, wildlife is publicly owned. We all own wildlife. The different countries in Central Asia and my collaborators, every country treats their wildlife differently. But at the end of the day, it's not my wildlife. You know what I mean? I have the skin in the game as the degree, but I am not the person who interacts with or benefits from or, is linked in a myriad of ways from other people. And so I feel like it's really important to just set up any kind of conversation you have from that perspective, particularly just because wildlife conservation in general has a problem with the Westerner savior complex. Which is a whole conversation that can always be unpacked. And so, like, you know, I think it's really important because I've been able to work with a lot of Chinese and Kyrgyz and Uzbek researchers. I work with people from all different 12 range countries, but I've been able to be in the field with people from all 3 of those countries. And it was completely bonkers. The first time I went out into the field, you know, you have to like physically train for it because we're talking about really, really high elevation. Like, I'm from North Carolina, like, I don't have enough oxygen in my blood to like just like run up the mountain. And so it involved a lot of training. And so like climbing stairs and just, you know, really, really hoping I didn't, you know, like falling off a mountain is not that big of a deal. Like, I'm not really worried about it. Like, I don't want to get eaten by a bear, and I just really don't want to get like high altitude sickness. The first time I went to China in the field in fall of 2018, I was there for 3 days. We were doing great. And like on the 5th day, it snowed, and that changed the barometric pressure. And I got really bad high altitude sickness. And it's not like, it can be dangerous. Like, in terms of, people can actually die from it because you either get like encephaly on the brain, or you get like a bunch of stuff in your lungs. And I couldn't get my heart rate down for about 24 hours. Like, my heart rate was about 110. And I wasn't like moving at all. Like, I was just like laying down. And so we got back to a city, and like my head was just really bad, and we agreed that I needed to leave. And so that really sucked because I was supposed to be in the field for like 3 weeks, and it was like my first time out in the field. And here's this, you know, this American chick like rolls up, and she's hanging out with you for like 5 days, and then she's like almost dying and has to go home. So that was hugely humbling and you know, awful. But once I trained a little bit differently, and I started taking, like, you take this cool medicine that helps your body change oxygen levels, or help you like process oxygen and carbon dioxide in your system. And so when I went back the second summer, I didn't get sick, which was great. But I mean, I don't even know how to like explain it. Being on the Tibetan plateau is a privilege that I don't know that I could articulate. I saw 4 snow leopards in summer of 2019 in person, like from like 300 feet away, because in this part of snow leopard country on the Tibetan plateau, the local herders, it's part of Buddhism. They have a high tolerance for wildlife, even when it involves like depredation events on their livestock, which is their way of life. And snow leopards are revered in many parts of the world, but certainly in China. And so these herders really lived alongside these snow leopards peacefully. And so, you know, this area had really high density of snow leopards, but the snow leopards just like, oh, hey, what's up? Like, you know, people in a van, like whatever, you just stay over there, like, don't bother me. But like, I'm just gonna do my thing. Like, think about Yellowstone, like, you know, when the wolves are just hanging out or the bison are doing stuff. It was like that with the snow leopards. And that was like the coolest thing in the entire world, because I got to watch a single snow leopard, the same female who had cubs. We didn't see the cubs, but we could tell she was lactating. She did 2 different hunts, like right in front of us. Like, she was trying to catch some blue sheep. She came like charging down the mountain, and it was snow. It was like this Nat Geo moment. She like jumped, she just like crested over the mountain and came charging down in the snow. And it was just like, you know, you could hear the angels singing kind of thing. And then like the next day, or like the next week, it wasn't snowing, and she was in this field of wildflowers. Like, the whole field was green, and there's these white and yellow wildflowers, and she's just like sitting in the wildflowers. And so that was like super magical. But aside from that, like, to answer like Jay's question earlier, I didn't get a chance to answer it. You're asking about like how it is to be like a woman in science, and like to take it down a notch. You know, there's always like a lot of layers to it, but some of the things that I've experienced, certainly in the field, is always like, I have been assumed several times to be the assistant on my own research. Like, when I was doing my master's, I was trying to collect tissue samples from coyotes at a predator hunt in Texas. And my now husband was helping me. And so he's like bad guy. He's like carrying stuff. He's like, what do I do? Like, I don't know how to cut up coyotes. By the way, they were not alive. Like, I wasn't doing surgery or hurting animals. And, you know, these like good old boys come up, and they're like, oh, hey, bud, like, you know, time to go do blah, blah, blah. What do you want to do? And my husband's like, she's in charge. And they're like, huh? You little, you know, kind of little lady thing. And that's inconvenient, obviously. But, you know, the thing that was, I think, the most surprising is that when I went to Kyrgyzstan in fall of 2019, it was on a National Geographic grant that I got. And so this is an early career grant that I got myself. I'm really proud of it. I mean, obviously, everything takes a village, right? Like, I I had help, obviously, like we all need help. But this was like my first grant that was my name, like a big grant on it. And so I'm really proud of that. And so I was in charge of everything, like the budget. I hired people, it's a team effort, but I still hired people. And like we get out into the field, and it became obvious the first couple of days, like they wouldn't let me hike by myself. And I was a little frustrated, and, basically like my Kyrgyz teammate who we wouldn't have been able to do it without him. He was really uncomfortable, but he was uncomfortable broaching the subject, but it basically became obvious. He said, we don't want you hiking alone like the guys do, because we don't know every herder in these mountains. And so I am literally at 14,000 feet above tree line, or about 14,000 feet above sea level. I am at the roof of the world, where the only thing you really need to worry about is like a brown bear. I'm looking for cat poop, and I have to worry about being harmed by a guy in the mountains. And so that was really sober. And I think, it was frustrating. I mean, we obviously adapt like we all do, but that was certainly frustrating. It's everywhere is I think the point. And so that sucks.
It sucks. You're like super. I'm listening to all your stories, and I'm just trying to picture them like they're beautiful. But that just shows that you persevere and you're a strong woman. So I applaud you and congratulations, because you've done a lot. Yeah, it takes a village, but yeah, definitely give you that hand clap of admiration there. That was yeah.
Your story just makes me think, like, it raises a lot of awareness for me, because I think about how nature is supposed to be for everybody. It's supposed to be free. It's the outdoors. It's free air that we all breathe. We're outside. We're supposed to be appreciating nature, and yet we still have these barriers to us really being able to appreciate that freedom of just being outside. And it's brought a lot of awareness to me. Thank you for bringing that to my attention, because now it makes me want to advocate for more safe spaces in nature, in nature exploration, conservation.
Backyard base camp.
Exactly. Bing, December, can you bring that up on the screen?
That was a good segue. Imogene, can you tell a little bit more about the altitude sickness? Is there immediate treatment if you move towards a lower altitude?
Yeah, so generally when you start to feel like it can come on really, really fast, it can be gradual, like it can feel just, like you've got a headache. It's like if you've ever had a tension headache, like where it feels like someone's putting a belt around your head and just kind of squeezing. It certainly can feel like that. Generally going to lower elevation will stop. It will reverse the effects of it. The problem is that, and I can't remember the actual metabolic pathway, but basically the longer you're in that high elevation when you're not handling it, it's like acetylcholine. There's something in like the ATP system, like the way that your body, it's effectively kind of like the same concept as building up lactic acid in your muscles. Whereas our body, like when we feel that burn when we're exercising, but when we stop, it quickly dissipates. The problem with altitude sickness is that after a certain point, you no longer get that dissipation in your bloodstream, and it's not lactic acid, it's something else. I can't remember, but yes, it can become problematic, and it either becomes a problem with like fluid on the brain or fluid in the lungs. And so generally, like what we do now is you have a medication that can help with that. It's called acetazolamide. If anybody wants to look it up, it's a super cool medication because it basically changes how your body processes the byproduct of not having enough oxygen. And so like when I went back the second time, I took it preventatively versus like taking it after. Like when you have a headache, you take Advil. But this is something that you take for several days beforehand to get your body used to it. And then I took it every single day that I was in the field for like 3 weeks at a time, just to make sure like I don't ever get those symptoms. So I'm not in danger. And so like all I ever got was a mild headache that second time.
It's great information. Amanda, I saw you turn off your.
Thank you. I was typing in our little thing to our producer. But there's a question in the chat again from Osama. How do you prepare to work not just in the field, but in different countries with distinct cultures, expectations, languages, etc.? In other words, can you share about the cultural competency preparation?
Right. Yeah. So that's a fantastic question. I think that's something that certainly we don't do enough of, and even like myself included, just because. Like going into another country, I am, whether it's in like in the city, like when I go to Beijing to do like lab work at like Peking University, I don't speak Chinese. Chinese is very difficult to learn, and so trying to pick it up is something that I've focused on. But in terms of being useful, I'm not. But when we go out into the field in China, the herders that we work with speak they speak Tibetan, and so a lot of not all the Chinese researchers speak Tibetan. So you usually there's like literally 3 different people. So you've got your Tibetan herders who know where the snow leopards are, and they help you navigate out in the field. And so they're speaking to the Chinese researcher who can speak Tibetan. The Chinese researcher is then either translating that directly into English, or the person who knows Tibetan maybe doesn't know English, and so then they have to translate it to Chinese, and then someone has to translate it to me, who's the most useless person there when it comes to language. And so that's certainly something that have dealt with in China. I am a little bit better with Kyrgyz just because the language is not so difficult. And when I say better, I mean I have like a list of like 20 words that I know, and only a couple of them are like grammatical sentences. And so again, like I'm just not as useful. And so the reality is, thankfully, a lot of the countries that I work in, or all the countries that I work in, English is taught as like a second or third language. And so somebody is going to know English. So in contrast, the United States, where we can't even like get our school systems to teach Spanish as a second language, which is completely ridiculous. We have students in all these different countries in Central Asia that speak Uzbek, they speak Russian, they speak Chinese. Sometimes it will speak English. Or if you're in China, you speak Chinese, you speak English, you speak Russian, you speak German. And it's something that is taught across generations. And so for me, one of the things that I'm working on now is since I've only been to 3 of those countries. I was supposed to go to India last year, but it didn't happen. Obviously I can't learn all of those languages, but I still think it's important to be respectful and make myself useful and know like basic words, like common courtesy words in all of those languages while I'm there, as well as like certain things like transactional conversations that are important. But the benefit is that a lot of the countries in Central Asia that snow leopards are in are former Soviet Union countries. And so a lot of the people all speak Russian. So like, Kyrgyz is their first language, or Tajik is their first language, but a lot of them also speak Russian if they are of a certain age. And so in terms of kind of trying to like check off a lot of boxes, being useful, if I'm not learning 12 languages, if I can only learn 3, like adding Russian to that is really important. But also talking slowly. I'm a fast talker. So when I'm when I'm speaking English, I have to be mindful all the time. Like you need to talk slowly. And you need to be extra courteous and extra polite because, you're the guest and they will treat you like a special guest, which is generally the way it is in Central Asia. They treat you kind of like a revered guest, but I am a guest, not a special guest. I'm a guest, if that makes sense. And so I think that is the best way that I have been able to navigate some of those differences. Faith is really important in Central Asia, particularly there's several different religions, but Buddhism is very important in certain parts of Central Asia. And I was invited into a 3,000-year-old Buddhist temple in China the first time that I went there, and I'm really private about that kind of stuff. I don't like go to church, but that was a really amazing, humbling experience. And it really just kind of shows you how connected, everything is, if that makes sense. I mean, it's just, like the United States is so young, like we don't even think about, like it's 3,000 years old. It was carved into a mountain. It was, it was a really humbling, amazing experience. And I think that coming from that perspective is the only thing you can do, particularly when you don't know so much, which is me.
That's so awesome. So, Imogene, we have one last question from the audience that we think is super important, and it comes from Clara. And Clara says, can you give any career advice for a graduating biology student about finding your balance between research and more popular science communication, or as we like to call it, SciComm?
Yeah, no, that's a great question. I think everyone here, and I think that it would be cool to open it up if you guys have anything to add. If you folks have anything to add, you definitely should. There are several different institutions and different professors and professionals who don't value science communication. They don't want to pay for it. They don't think that you should be doing it. You're a grad student. Why would you be focusing on like layperson education, or why would you be focusing on someone else's education when you can only be focusing on your own? And like my answer to that in our field is that in terms of conservation, because conservation is about people, you're only going to get people to love something if they understand it, or if they know it, or if they appreciate it, if it comes to them in a way that is intriguing to them. Because what's cool for me might not vibe with you. And so like we talk about it in a casual way, like not this, pie in the sky, like high on our horse, like ivory tower academic. We're way more to find common ground, or we're way more likely to find common ground. And so with that in mind, I think that the balance is really about juggling your time, but also not being apologetic about how you choose to spend your time. So initially when I started, yes, SciComm was like mostly in my downtime. Like I'm working on my master's, I'm working on my PhD, like this has to happen after hours, or if it's happening during the day, like I have to work extra hours. And certainly that still happens. But if science communication occurs in real time. And so if you only treat it as something that you do after hours, like on the weekends or at the end of the day, you're not going to reach your audience. And so I think the reality is finding a way to link it back to your work. So even though I do science communication, I have gotten a lot of cool connections. I've been able to go, I've like found out about educational conferences to teach me more through Twitter. I'm doing science communication in general on Twitter, but I've met people, I've found papers that have been relevant to me and helped me chase down like programs that I need to learn about and given me new ideas. And so it's always come back to that. And so I'm better able to kind of justify it to those like higher ups who don't necessarily value it. But what it does is it proves, yes, this has a place in our professional world, not just as like a fun hobby. And so that would be my recommendation, is to try to find a way to make it relevant to your work as you also work for other people. I don't know, what do you folks think?
I mean, we met on the internet. Like we met through Raven's Instagram account, and like we're all pretty active on Twitter. So it's like you're very right in saying like that is like it's where the people are. If you're gonna talk science, if you're gonna communicate, you gotta go where the people are.
Right? Yep.
Yeah, 100%. Yeah, I definitely agree. So my dissertation's in science communication, and I'm learning that the field of SciComm, although it's adjacent to science education, it is a different field. It's a different way to practice or educating about science. And there aren't many like formalized programs around science communication. In particular, my experience trying to get opportunities to blend my interests in formal biology and science education even have been stunted by the culture of STEM right now. That's not so accepting, as Imogene was saying, of scientists who are really great at being in the lab and being at the benchtop, also having interest in serving the community and actually caring about something else that's not at the benchtop. And that's not to say that that's every institution. There certainly are institutions that are accepting, that are welcoming, but I didn't access them, based on my thorough opportunity search. I still didn't come across anything. And I was actually rejected from biology PhD programs because I expressed that I was interested in the kind of work that I'm doing today, which, actually ended up getting an apology letter from one of the programs that rejected me because they realized that they were mistaken, which is fine. Yeah, but I mean, ultimately, I think I say all this to say many of us are still kind of forging our own paths, right? I don't even think I was on ASAP Science's, we were recording a podcast, and they were like, we all just agreed that we didn't even know what science communication was as we were forging our path in SciComm. We didn't know there was a name for it. We didn't know what we were doing. We were kind of just like going with our gut. Like we just want to help people learn about science in a fun way, in a way that reaches more than just our inner circle or people in our lab. And we just kind of followed our heart, So Clara, if there's something like that you feel like is really calling you. Like if there's a cause, if there's something you want to bring awareness to, maybe you're really passionate about diabetes awareness, or maybe there's a certain demographic of people that you feel like are traditionally left out of science that you want to reach. Like follow that instinct and just understand that that is a part of your calling. You know, it's not guaranteed that you're going to get opportunities to do SciComm from the institutions that you are involved in, but like don't let that scare you away from not doing it. You have a huge network like on social media. Just like reach out to people, ask, can I get involved in your thing? You know, my school's not really cool. They're not with it yet, but like let me know how to get involved. And people will definitely be sure to help you out. And there are professional organizations for science communication that you can also get involved with as well if you just need a little help finding out how to get opportunities as well. So let us know. Keep watching STEMbassy. Like we'll be looking for your name, Clara, and looking for updates to see how your SciComm journey is going. So don't forget about us because we want to hear from you again.
Well said.
Awesome. So is it time to transition into our special segment?
Public health hat time?
And for the public health.
Is it time public health hat?
We seriously need a jingle.
We
We
do.
do. We need a jingle.
Season 4.
Season 4, we need a jingle.
All right, you ready? It's gonna be you time.
Okay,
All right.
ready. Okay, guys. So tonight's public health hat is brought to you by Miss Verona. First, she told me to tell y'all, she says, hey, so hey, y'all. Okay, so back to the message. With the onset of mass vaccinations occurring across the US, you would think that the incidence rates would be decreasing. But my fellow Americans, quite a few of you have been adamant about going outside without not wearing a mask, attending social gatherings, and having parties in your homes with close friends and family, and even pressuring your local governments to reopen the states and lift capacity limits on indoor dining. Yep, y'all did. Well, guess what? Over the past 2 weeks, incidence rates for the coronavirus have increased. That means they've gone up, even on the verge of getting everyone vaccinated or lots of people vaccinated. You can check the link that we're posting really soon. It should be posted here really soon. It's from NBC. And it was updated as of 7:41 p.m. before we started the show tonight. But you can see the percentage of rate increase of incidence rates in your area, the coronavirus positive test in your state via that link. Okay, all that being said, listen, vaccine or not, that takeaway message here is to stay away from each other until we get a handle on this thing, okay? You can't be out here partying. You can't be out here with your friends and your family that you don't live within the same house because we don't have a handle on this thing yet. It's spreading all across the nation. Now, Ms. Ronaldo says she got a free trip from West Coast to East Coast, and she is very happy to do it again, make another round. She's very happy about this. If you don't believe her, check the rates in your state, okay? Now, just in case you didn't get the message, here's the message. Okay. Make good choices, y'all. Make good choices. And that is this week's public health hat. Have a good week. Make good choices.
Where can we get this shirt?
Yes, I need one, please.
So this
I
shirt is courtesy of the good sis at Mess in a Bottle.
All right. Thank you, sis. We'll
Mess in
be
a Bottle. That's short for Message in a Bottle. And when you get it, when you order from her website, you actually get your mess in a bottle.
I love it.
Oh,
I love it.
You're
Thank you.
welcome.
that's awesome.
I love that. That's so cute.
That's a great public
Public health
announcement.
hat.
Yeah.
It is.
We love a public health hat.
Chanel, real quickly, can you address the people that says that the virus is not here, like it's gone? I've heard that a lot lately, like it's gone.
So
Yeah.
without getting into nuances or playing the blame game or getting into any of those things, what I will say is this. If you don't believe that the vaccination is a good idea, if you don't believe that the coronavirus is real, I would just ask you to be very sensitive and empathetic for those of us who have actually lost loved ones due to the coronavirus pandemic. I myself have lost loved ones because of that. And in actuality, the loved one that I lost was actually through secondary exposure. So someone in the household actually brought it to the household. The entire household came down positive with COVID, and unfortunately, one of them did not make it. So I think that whether you believe it's real or not, that is a personal decision. But I think that we all need to have some empathy and be sensitive to those of us who have lost loved ones due to the virus. And in the meantime,
right. Thank you.
Just saying. You don't gotta believe it, but can you at least do this? This is free. Don't cost you nothing. It's free. Now the shirt gonna cost you a little bit, but the concept is free. It's free.
Kindness
Stay
is
6
free.
feet away.
I love that.
Darn good answer, by the way. Darn good answer. I mean,
Science for the community. You know, you gotta give the people the way they are, where they are, and how they are. You don't gotta believe it, but at the least, stay away. Thank you.
Some people will only understand if you say it like that.
I think so.
Be like, I said
What I said. Okay.
what I said. Okay, exactly how I said it. Okay, right.
I mean it.
I had a expandable yardstick that I kept in my purse, and I would just casually I would just so casually, when I was at the store, scrape it out my bag. You know, I wouldn't say nothing at first. I just want you to see I have it. You know, first of all, I want you to see I have it out, ready for you.
don't make me pull it. Especially.
And then I'd like straighten it out a little bit, and people be like, "Oh, you know, back up, back up."
And you make them hold the other end, or you just whip it out.
You know how you take it out and it doesn't straighten. It's like the metal ones is bendy, but if you like fling it a little bit, it straightens out.
Next time, make them hold the other end. Hey, can you hold this for me? I just want to see how far. Just want to know. Like 2 inches? Okay, back up.
Have you heard of 6 feet?
But thank you. Exactly. Exactly.
That's so great.
Listen.
Well,
We got a segment.
yes, we do. Thank you, Jay. So we're going to do a conservation corner tonight with me. And so we're going to talk about something that is important for springtime. And so if you guys haven't heard, there is an insect called the spotted lanternfly. And so if you're on the east coast of the United States, it's about that time of the year where you're going to start seeing them again. December, can we pull up the image of what these spotted lanternflies look like? Yes, thank you. So you can see on the tree in A, that's what their eggs look like. B and C, those are kind of juvenile forms. And then D, that's how the adult looks with its wings closed. And E, that's how it looks like with its wings open. And so this is an invasive insect to the eastern United States. It was found in Pennsylvania in 2014 and has spread widely in eastern U.S. And it is continuing to spread, and it is important because it is affecting our industries like orchards and other fruit crops. What happens is they excrete this substance that ends up bringing like rot and fungus to the crops, and it can completely wipe them out. And so it's not really good for our agriculture here in the United States. And so when you see an egg mass like in A, or if you see a nymph in B or C, or one of the adults in D or E, go ahead and squish it. Like you have permission. Like it's not supposed to be here. It is not part of our ecosystem. Our ecosystem hasn't evolved to be able to deal with it. So we need to try and remove it. And so if you see one of these, go ahead and squish it, put it in a little baggie, and send it to the agriculture department in your state, especially if you're in a state that hasn't seen it before, because it's important for them to track if it's coming or going anywhere. Also, make sure you're looking on your cars to see if you see egg masses like an A, because this guy likes to hitchhike. It's going to lay eggs on your car, in your undercarriage, and it's going to try and cross those state borders. And that's how it's been spreading throughout the eastern United States. So if that happens, what you can do is you can get like a car scraper, and you can scrape the egg mass into a little plastic baggie, and you can squeeze some hand sanitizer on it, mix it up, seal it up, and toss it out. And the hand sanitizer ends up killing those eggs. You could also use like rubbing alcohol as well to kill it. And like I said, just make sure that you report it to your local Department of Agriculture, especially if you're in a state where it has yet to be seen. And so you can look at the USDA website, APHIS, and I can try and get a link for Dee to post while we do our closeout, and you can see if it has not been reported in your state yet, and it also gives you contacts for if you do need to report it so that the Agriculture Department can get on top of it. So that's our conservation corner. It's just about an invasive species that's trying to wreak some havoc on a lot of our food crops, and we need our food availability so that people can eat healthy and survive. So yeah, that's Conservation Corner, and I'm gonna look up that link for us to post really quickly.
Ariana, that egg mass was so interesting. It looked like tree bark.
Yeah, they're sneaky. They do kind of camouflage and look like tree bark, and so you can kind of see it, and it will scrape off. So if you do see it, it should scrape off pretty easy.
Oh my God.
Yeah.
Just squish them.
Yep. Squish the adults, squish the nips. If you see the egg masses, scrape them off, smush them, put them in a bag, put some hand sanitizer on it. So here it is.
I wasn't expecting squish them to be the instructional activity of the night, but okay, I will. If you say I can, I will.
Imogene and I, before last year or 2 years ago, I can't even remember now. Time runs together. We were walking on Main Street here in Newark, Delaware, and they were everywhere. And Imogene and I were like running up the corners and just like stomping on them with our boots because they were just littering the streets. They were.
People thought something was wrong with us. Like 100%.
I'm protecting nature, okay? Like,
make it make sense.
Basically.
Yep. So that was our conservation corner. Maybe I'll do more of these in the future if we like them. So
yes. I love learning about. I remember when I first moved here, one of the biology professors was really into making sure we were aware of like the natural flora and fauna of California, and like I come from Florida, so it's kind of cool to be like, oh, well, this is like the natural wildlife and like the plants that are native to this area, and this is what isn't supposed to be here. And it was interesting to just like find all that out through something that was like, I don't know, around me.
Right.
Well, this has been so much fun, Imogene. Thank you so much for your time. If you all want to hear and see more from Imogene, you can check her Twitter at biologistimo or IG at biologist_Imogene. And this week we were fundraising for Backyard Base Camp. And you can check them out at backyardbasecamp.org. If you got some extra coin and you feel like donating it to a good cause, go there, click on the donation link. You know how to do it. You can also find STEMbassy at STEMbassy.org, on socials at STEMbassy, on Facebook at the STEMbassy, and on patreon.com/STEMbassy. That's all that we have for tonight, and STEMbassy is adjourned. It was good seeing everyone.